Hello everyone. In this video, we learn more about the polymerase chain reaction, or PCR. For a long time, the only way to amplify or obtain multiple copies of a gene was to clone it into a plasmid, insert the plasmid into bacterial cells, and allow them to multiply. The entire process can take up to 3 days. Genes can now be amplified in a couple of hours using polymerase chain reaction, more popularly known as PCR. PCR was developed by Dr. Carey Mullis in 1985. He won the 1993 Nobel Prize for his invention. If you want to learn more about Dr. Carey Mullis, you can read his book called Dancing Naked in the Minefield, which talks about his views on various topics ranging from HIV to climate change. PCR takes place in a machine called the thermocycler, as shown here. This is where the samples go, and the samples are contained in tiny tubes called PCR tubes. PCR involves a series of heating and cooling steps that can be programmed into this PCR machine. There are just a few reagents needed to set up a PCR reaction. The toolkit involves the DNA to be copied, which serves as a template. So if you have a DNA sequence and you want to copy part of it, you need to have the DNA that contains that target sequence. The next thing you need are DNA primers. These primers are short sequences of DNA that will bind to the ends of your target sequence. Next, you need a DNA polymerase. PCR uses a special kind of DNA polymerase called the Taq DNA polymerase, which is isolated from the bacteria Thermus aquaticus, which live in hot springs in the Yellowstone National Park. The reason this polymerase is used is because it can withstand high heat. You also need dNTPs, or nucleotides, that will be added by the polymerase to make new strands of DNA. Lastly, you need a buffer solution in which the reaction can take place. This provides a conducive environment for the reaction to occur. Let's look at a general overview of the process, and then I'm going to take you to a website that offers a visualization of this process. To begin with, present in the reaction is the DNA template that contains the target sequence to be copied, which is right over here. When you raise the temperature of the reaction to approximately 95 degrees Celsius, the double strands separate because DNA melts at that temperature. The hydrogen bonds between the base pairs are broken. The temperature is then brought down, which allows the primers, which are present in much more quantity than the DNA template, to anneal or hydrogen bond with the ends of the two strands of DNA. This step is called primer annealing. Note that this is all happening in the first round of the PCR reaction. Next, the Taq polymerase works at an optimal temperature of 72 degrees Celsius. So the temperature of the reaction in the thermocycler is raised again to this temperature, which allows the Taq polymerase to extend and make new DNA using the nucleotides or dNTPs that were added to the reaction that are complementary to the sequence in the template strands. This completes one cycle of the PCR reaction. In the next cycle, the same steps are repeated: denaturation, primer annealing, and primer extension, giving you 4 pieces of DNA, which vary in length, but none of them contain just the target sequence that we want. That target sequence is achieved after multiple rounds of the PCR reaction. So in the next cycle, you can see that there are 2 copies of the target sequence that we have. If you run this reaction many more times, you would increase the number of the target sequence exponentially in the reaction. I'm now going to take you to a website that offers a visualization of this process. I've been using a lot of information from this website as a resource for the course. It's great. So let's begin. The human genome is made up of 3 billion chemical base pairs. Scientists often need to isolate pieces of this very large genome. In order to do that, they need to have multiple copies of it. Going into the cell nucleus, you have the DNA, and if we want to look at one part of that DNA, we would need to use the polymerase chain reaction. And as Dr. Cathy Mullis described the technique, it requires no more than a test tube, a few simple reagents, and a source of heat. So our aim is to multiply that segment of DNA. This DNA can be extracted from cells, and these cells can be any cells from the body. The advantage of the technique is that you don't need much of the starting sample because the whole goal is to amplify the DNA. So we're going to start by moving our DNA template from a larger test tube to a PCR tube. Note that all of these tubes are placed on ice to keep them in the appropriate condition and not allow them to react with each other before they need to in the thermocycler. So let's go ahead and do that. Okay, that's done. Next, we're going to add our first primer, which is complementary to one end of the DNA segment. Now, before I release this, note the tip on the pipette. This tip is going to be discarded after every transfer to avoid contamination between the samples. Now we'll add the next primer. The next thing we'll add is the nucleotides. These are A, T, G, and C that are normally present within DNA. And lastly, we'll add the DNA polymerase called Taq DNA polymerase or just Taq polymerase. Now our reaction is ready. Note that we've used all of 5 reagents for this one reaction. So what we're now going to do is transfer the reaction to the thermocycler. All that the thermocycler does is fluctuate the temperature of the reaction. So we're going to start our first cycle. Zooming into the tube. Target temperature now is 95 degrees Celsius, and we've zoomed in on our target sequence. At this temperature, the DNA is going to separate into its single strands. The temperature is now reduced to 50 degrees Celsius, which allows the primers to anneal or bind to the DNA template, specifically the target sequence. The temperature is now raised to 72 degrees Celsius, which allows the DNA polymerase, since it works well at this temperature, to copy the template strands and produce a new DNA sequence. Now, cycle 1 is complete. We're going to go on to the next cycle. The same 3 steps are going to be carried out again. Temperature lowered, primers attach, DNA polymerase copies the segment. And the temperature is now going to be raised for the 3rd cycle. Okay. At the end of this 3rd cycle, you see that there are 2 pieces, like I said earlier, of just the target sequence. Now, in order to make more of these target sequences, we need to carry out more cycles of the reaction. So now we have 8 pieces of the target sequence. At cycle number 5, we have 22 fragments that contain only the target sequence and only 10 longer length copies. So we have much more of the target sequence than we have the longer copies. Now, if we fast forward and carry out multiple steps, say 30 cycles, much more of the target sequence than you have the longer length copies. And this is the sequence you want. This is the target sequence you want. You can use these multiple copies of the target sequence for various purposes, ranging from amplifying a small DNA sample that you obtained from blood at the crime scene, or to make multiple copies of a gene that scientists are studying in the lab to figure out the sequence of that gene. So there are multiple applications of this one technique, which is why this technique is popular in laboratories around the world. Thank you.